Welcome to the Leaving Cert Biology channel. This ties hand in hand with our website leavingcertbiology.ie, which is a platform for 5th and 6th year students in Ireland studying Leaving Cert Biology. Of course, we have our TikTok and Instagram page at leavingcertbiology.ie as well, where we offer completely free videos, tips, tricks, exam strategy and trends. And on our website, we have exam paper structure, timing, all of our completely free podcast episodes, and so much more. Our aim is to support students all over Ireland. Okay, so in this video, highly requested, we are going to look at Unit 2 Genetic Crosses Part 1, and we're going to focus on monohybrid crosses. But before I do any of that, I am going to talk about the genetic crosses that you are required to know. So the first one, which we do today, is monohybrid, one trait, dihybrid, crossing two traits, incomplete dominance, pedigree crosses, and linked or sex linked. We're going to start off by talking about all of the definitions that you could be most commonly asked in a Leaving Cert Biology paper, and then I'm going to get into doing a monohybrid cross. So definitions, let's start with gene. A gene is a section of DNA that codes for a particular protein. Autosomes are the first 22 pairs of chromosomes, and pair 23 are your XX or XY sex chromosome. The homologous pairs of chromosomes are called karyotype, and heredity is the passing on of traits or features from parent to offspring by means of genes. Locus is the position and location of a gene on a chromosome. Alleles are different versions of the same gene. Dominant is stronger than the recessive. It is expressed, preventing the recessive from working, represented by a capital letter. Recessive allele is stopped from working if a dominant gene is present, always represented by small letter. Homozygous when the two alleles are the same, and heterozygous when the two alleles are different. Then we have genotype, the two letters used to represent the genes, and phenotype, I always think PH for physical, so the physical appearance of the organism. Progeny is another word for offspring, and sex-linked are genes located on the X or Y chromosome. Linkage, two genes occurring on the same chromosome, and incomplete dominance, neither dominant nor recessive allele is completely dominant. Okay, so let's start off with our genetic cross steps. First thing to note is that layout is really important. Don't get too good at doing these. Keep your headings because it allows your examiner to follow your work. Then if you make a mistake, your examiner will be able to follow. However, if you have no headings, it's a lot harder for your examiner to follow and give you marks. So step number 1, write out your heading for parental phenotype. In other words, what the parents physically look like. Remember, PH for phenotype, PH for physical. Number 2, write out the parental genotypes, so their allele combinations, the letters, okay, underneath. So what do they actually look like? Are they homozygous? Are they heterozygous? Then we have number 3. Check if the question is going to mention incomplete dominance or sex-linked disorders like haemophilia, colour blindness. So that's just a yes or no. It will be something you'll have to be aware of. And if the question does mention it, then you can continue as normal. Number 4, gametes. So each parent can only pass on one gamete, okay, to offspring. 5 is do your cross, also known as your Punnett square, and 6 is to write out your offspring genotypes, known as your progeny or your F1 progeny, and then your F1 phenotypes of your progeny, what they physically look like. So here we have an example of a monohybrid cross. Let's look at the question and let's underline what we need to know. So in peas, green seed, capital G, is dominant to yellow seed. And we're not talking about anything else, only seed colours, so we know that this is one trait. So I highlight that green seed, capital G, is dominant, and yellow seed, small g, then is obviously recessive. A pea plant that is homozygous for green seed. That homozygous means the letters are the same. And because it's for green, it means we're going to have two big G's. And that's going to be crossed with a yellow seeded plant. Now there's only two combinations of G's that will give me yellow, there's only one combination of G's that will give me yellow, and that is two small G's. So step number 1, write out your parental phenotypes, what they physically look like. So we are told that we have a green parent crossed with a yellow parent. Okay, so a green plant with a yellow plant. Okay, so remember, homozygous for green is crossed with a yellow seeded plant. On to number 2 then is to write out the parental genotypes, so their combinations of their letters. Now we are told we have homozygous for green, and that green was dominant. Homozygous means the alleles are the same, so that means I have 2 capital G's, okay? Homozygous, dominant. Okay, so be very careful with that. Remember, the opposite of a homozygous is a heterozygous, and this is when the letters are different. The other plant that I have then is a yellow seeded plant. And remember, small g for yellow is recessive. So if I'm to have a yellow seeded plant, the only combination I can have is small g, small g. So one parent is big G, homozygous dominant. The other parent is small g, homozygous recessive. And then we move on to step number 3. Does the question mention incomplete dominance or sex-linked disorders like colour blindness or haemophilia? No, it does not. And then number 4, we write out the gametes that each parent can pass on. Remember, each parent can only pass on one allele to their offspring. So parent number 1, green, has 2 big Gs. The only gamete that can pass on is a big G. Now you can of course write it twice if you want, and small g, yellow parent can only pass on a small g. Again, you can write it once, you can write it twice, either is completely acceptable. Step number 5 then, you are going to do your cross or your Punnett square. So we have our parents, we have their gametes. I always say that one parent's gamete goes down the side and the other goes along the top. So I put the big G gamete from one parent down along the side and the small g gamete from the other parent along the top of the cross. And then we're just going to cross them. So a big G with a small g, always write your dominant letter first, and that's going to give me big G, small g. And there is no other combination that I can get from that cross. So big G, small g, step number 6, we write out the progeny or the offspring genotypes, which is that big G, small g. Now, then for step 7, we have to write out the progeny phenotypes, what they physically look like. Because I have the dominant allele, big G there, it means that heterozygous, the dominant is expressed, prevents the recessive from working. So we have 100% green offspring. So remember, just a note, exactly as I said there, in the heterozygous, in other words, like our step 6 there, when the alleles are different, we have the dominant present. And because we have the dominant allele present, it prevents the recessive allele from being expressed. So what our offspring will look like is the dominant. Now, in a nutshell, that is a monohybrid cross. Obviously, you can be given 2 heterozygous parents, a heterozygous with a homozygous, 2 homozygous dominant, or 2 homozygous recessive, but have a practice for yourself. If this booklet is of interest, it's available on my website with a whole load of worksheets compiled within it on leavingcertbiology.ie. If you want to see a part 2 with a dihybrid or a pedigree cross, please let me know. As always, thanks so much for watching. Hit the like button if you enjoyed or comment below, and I will talk to you all in the next video.